So in order to talk about the role of novel agents in immunotherapy, I'll just quickly highlight why we're considering novel agents and how we came here. Well, primary CNS lymphoma in the past has been thought to be quite indistinguishable from systemic lymphoma, only aggressive and in the brain. And on histopathology, it looks quite similar to a systemic diffuse large B-cell lymphoma. However, now upon comprehensive genomic analysis, we know that this is a distinct disease with unique targets. Primary CNS lymphoma exhibit increased toll-like receptor signaling as a result of MYD88 mutations and increased B-cell receptor signaling as a result of CD79B mutations, which occur concurrently with MYD88 mutations. And so we see increased NF-kappa B activity in primary CNS lymphoma and several targets along this pathway that could be targeted. The most studied pathway and target in primary CNS lymphoma has been BTK, and targeting BTK, or Bruton's tyrosine kinase. So the first study was ibrutinib, which showed a remarkable response rates between 50 to 77% in the initial monotherapy studies. However, the challenge with that was the durability of the responses. Even though we saw responses, we saw that there was the lack of durability and patients would progress pretty quickly. Since then, there have been additional studies with the next generation BTK inhibitors. So recently we conducted a study of, in the US of the second generation BTK inhibitor, Tirabrutinib, in 44 patients, where we saw that the overall response rate was 67% and the complete response rate was 44%. Importantly, in this study, we noted that the duration of response was over 9 months, which was really impressive, and the median progression-free survival was 6 months. There was another additional similar study with another next-generation BTK inhibitor, Zanubrutinib, that was conducted by the cooperative group in the UK, which also showed a response rate of 55% and a complete response rate of 35%. Again, a median progression-free survival of about 6 months or so. So BTK inhibitors in the relapsed refractory setting seem to be very promising. There is now the plan for a randomized phase 3 trial of Tirabrutinib versus standard of care chemotherapy with rituximab and temozolomide in the relapsed refractory primary CNS lymphoma population, which will hopefully be the confirmatory to our initial results. So in terms of targeted therapy, BTK inhibition seems to be really show us impressive responses, but also what we're trying to do is move it into the frontline setting simultaneously, because it's better to try to get the disease earlier on to prevent more and more neurologic damage. And then speaking of immunotherapy, we have definitely looked at the role of checkpoint inhibitors, but what is more interesting right now is the role of CD19-directed CAR T-cell therapy. Some of our colleagues and I conducted a study of Axi-cell, which is CD19-directed CAR T-cell therapy in relapsed and refractory CNS lymphomas, and this showed impressive response rate of 89%. But again, the durability was what was striking to us. In our study with Axi-cell, the median progression-free survival was 14 months, which is longer than what we have seen with any other therapy so far. There are other CD19 or CD20 directed CAR T-cell therapies that are being investigated, one of them being Zanubrutinib, which is also being investigated in primary CNS lymphomas, and additional results remain to be seen in terms of its durability of response. So certainly, there is a lot of interest from pharmaceutical companies to explore the role of novel agents and immunotherapies in primary CNS lymphomas. And I think what we've shown over the past years is that the concern that primary CNS lymphoma patients would probably have more side effects are not really truly seen in the context of these trials. And even with, for instance, cellular therapies, we showed that the ICANS rates, or the CRS rate, neurotoxicity, which was most concerning for people, especially having a brain tumor, have not been more than what has been seen in the systemic diffuse large B-cell lymphoma population. So I'm hoping in the future we can really move some of these therapies in the frontline setting and really get to curative options for our patients.